Assalamu alaikum students. Hope so you all will be fine. Students, I am going to discuss about the metabolism of very low density lipoprotein and low density lipoprotein formation. Metabolism of VLDL and LDL formation. Students, we will cover this topic under the heading of site, various steps for the metabolism of VLDL. Third one is uptake of LDL, and fourth one is disorder. Students, we will cover this topic under heading of site steps, uptake of LDL and disorders. The VLDL, uh, the site for the synthesis of VLDL is liver. The site for the synthesis of VLDL is liver. Okay, students. Basically, there are some complex of lipids along with apoprotein that gets synthesized in liver. Triacylglycerol, cholesterol, and ApoB100. Okay, students, triacylglycerol, cholesterol, cholesterol ester, phospholipid, these are the lipids, and along with this protein, they form a complex, and such type of complex gets synthesized in liver. Liver. And we are going to discuss about the various steps for the metabolism of VLDL. In next step, there is a synthesis of nascent chylomicron, nascent VLDL. In next step there is a maturation. ApoC2 and ApoE from HDL, they gets incorporated in nascent VLDL and leads to the maturation of VLDL. And leads to the maturation of VLDL. This is the mature VLDL. In next step, there is an important enzyme lipoprotein lipase which is present on capillary wall and it is fixed to endothelium by heparin sulfate. This lipoprotein lipase enzyme, activated by it is activated by ApoC2 protein and it is inhibited by ApoA2 and ApoC3 proteins. This lipoprotein lipase enzyme, it causes the breakdown of this triacylglycerol molecule and converts it into free fatty acid molecule that utilized in peripheral tissues, glycerol molecule which is taken up by liver for further metabolism, and there is loss of apoC2 protein and it converts into VLDL remnants and it leads to the reduction in the size of VLDL. Triacylglycerol, cholesterol, there is a ApoB100 protein. Okay, students, this is the VLDL remnants. It is also called as IDL, intermediate density lipoprotein. I am repeating again. This triacylglycerol, cholesterol, and various other lipids they form complex with these proteins ApoB100, and this complex gets synthesized in liver. This is the nascent VLDL formation. From HDL, ApoC2 and ApoE protein gets incorporated in the nascent VLDL and leads to the formation of mature VLDL. Then next step, there is a lipoprotein lipase enzyme which is activated by ApoC2 protein and inhibited by ApoA2 and ApoC3 protein. This enzyme acted upon triacylglycerol and converts into glycerol, which is uptaken by liver for further metabolism process, and free fatty acid tissues, which is utilized in peripheral tissues, and it converts into these VLDL remnants. It is also called as intermediate density lipoprotein, and it also loses apoC2 protein. In next step, there are two fates of this VLDL remnants or ideal. First one, uptake by the receptor mediated uptake by liver by the process of receptor mediated endocytosis or LDL formation. So in next step, what happens? See, this this intermediate density lipoprotein, it converts into the formation of LDL, having cholesterol and ApoB100 protein. There is in this way LDL formation takes place. Students, this intermediate dense lipoprotein having two fates. It is uptake by liver by receptor mediated endocytosis, or it leads to the formation of LDL. The third step is uptake by liver, uptake of LDL by liver. Okay, students, I am repeating again. There is a nascent VLDL formation. There is the incorporation of apoC2 protein ApoE in nascent VLDL. It leads to the formation of mature VLDL and the rejection of lipoprotein lipase enzyme, and it leads to formation of VLDL remnants, which is leads to the formation of this LDL, low density lipoprotein. And next, this LDL it is uptake by the liver. Students, this LDL is uptake by liver by clathrin mediated endocytosis. By clathrin mediated endocytosis. And this LDL it is uptake by which type of tissues? This LDL is uptake by only peripheral tissues and liver. Only peripheral tissues and liver. So students, now coming to the uptake of LDL. Here is a phospholipid And here is, this is the LDL receptor. The LDL is taken up by this receptor, LDL receptor. This is the, it is outer protein part, it is transmembrane protein, and this is the inner protein part. This outer protein part, basically it is a LDL attachment site, and this is the Clathrin attachment site. Now coming to the mechanism of this LDL uptake. Students, this is the Clathrin and this is the pit. We can say it is a Clathrin coated pit. Clathrin coated pit. And this is the LDL receptor. This is the LDL receptor, and this is LDL particle. What happens that this LDL particle gets attached with this LDL receptor and forms a complex. In next step, there is the internalization of Clathrin coated pits with the help of Clathrin. I am repeating again. This is the Clathrin coated pit. This is the LDL receptor, and in red color, this is the LDL particle. The LDL particle gets attached to the receptor and form a complex. In next step, there is the internalization of Clathrin coated pits by Clathrin. Students, in next step, and this is the formation of endosome. In next step, there is the shedding of Clathrin and receptor part reutilized again, and there is the reduction in the site of endosome. There is a reduction in the size of endosome. This Clathrin, there is a shedding of Clathrin, receptor part is reutilized, and there is a reduction in the size of endosome. In next step, there is the fusion of lysosome. There is a fusion of lysosome and leads to the formation of endolysosome. Now this endolysosome having three fates. It having three fates. It is leads to the re-esterification of cholesterol by acyl cholesterol acyl transferase enzyme, or it leads to the synthesis of plasma membrane, steroid hormone, and ketone bodies. And third one, it leads to decrease in the synthesis of LDL particle, LDL receptor, and also inhibition of cholesterol biosynthesis. Students, I am repeating again. That LDL it is uptake by peripheral tissues and liver by receptor by clathrin mediated endocytosis. This clathrin is it is a protein which helps in the endocytosis of LDL particle. Now this is the general structure that this is the LDL receptor which is made up of this outer protein. This is a transmembrane protein. This is the inner protein. The inner protein it is the clathrin attachment site. The outer protein it is the LDL attachment site. Now this is the mechanism. These are the clathrin coated bits. Inside there are LDL receptor and this is the LDL particle. This is the LDL particle and this is the LDL receptor. Okay, in next step there is a internalization of clathrin coated bits with the help of clathrin, and in next step there is a shedding of clathrin and receptor part is reutilized again, and there is a reduction in the size of endosome. It leads to the formation here. This is the next step. Shedding of clathrin receptor part is reutilized and there is a reduction in the size of endosome. There is a reduction in the size of endosome. And next step is a fusion of lysosome and leads to the formation of endolysosome. And in next case, there are three main fates of this endolysosome: re-esterification of cholesterol by acyl cholesterol acyl transferase enzyme. It leads to the synthesis of plasma membrane, steroid hormone, and ketone bodies, and it leads to decreased production of LDL receptor or inhibition of cholesterol biosynthesis. And this way, I have cleared three main steps of the metabolism of VLDL. First, synthesis. First is the site. Second one is the metabolism of VLDL, and the third one is the this uptake of LDL by clathrin mediated endocytosis. Now coming to the disorder of the this metabolism of VLDL LDL formation disorders. Students, disorder. There is a type 2, type 3, type 4 hyperlipoproteinemia, and same way here. The type 1 hyperlipoproteinemia it is due to the deficiency of it is due to the deficiency of LDL. The type 3 hyperlipoproteinemia it is due to deficiency of IDL, while type 4 hyperlipoproteinemia it is due to deficiency of VLDL. So these are the disorders related to this metabolism: type 2 hyperlipoproteinemia, type 3 hyperlipoproteinemia, and type 4 hyperlipoproteinemia. This type 2 hyperlipoproteinemia it is due to the deficiency of LDL. Type 3 hyperlipoproteinemia it is due to deficiency of IDL, and type 4 hyperlipoproteinemia it is due to deficiency of VLDL. So students, we have discussed here the metabolism of VLDL and LDL formation. First, the site for the synthesis of VLDL is liver, and second is the metabolism. First one, nascent VLDL formation. Second is the maturation. Third one is the LDL formation, and the fourth one is the uptake of LDL particle by the clathrin mediated endocytosis and various disorders. Keep watching. If you have any query, you may ask in comment section. Thank you.